Hello, this is lecture number 22 in the series from the 7th edition of Principles of Biochemistry by Albert Lehninger. Today we're going to begin the pathways of biochemistry. We will start with the one that's generally presented first in any review of biochemistry educational material, and this is the pathway of glycolysis and the reverse process of gluconeogenesis and the pentose phosphate pathways. Glucose in the body has four functions. First is an extracellular matrix and comprises part of cell walls as it bonded to protein. It also is converted to glycogen. It also occurs as starch and sucrose. You can oxidate it via glycolysis, and you can oxidate it via the pentose phosphate pathway, which gives a higher energy yield. In glycolysis, glucose is transformed ultimately to two pyruvate molecules. In fermentation, you get anaerobic transformation of glucose to energy. And glycolysis is in two phases. The first phase is glucose being converted to glyceraldehyde 3-phosphate. And then the second phase, glyceraldehyde 3-phosphate is converted to pyruvate. So let's look at the overview of this pathway for our own edification. We begin first with glucose, the structure of which is here, and then an enzyme hexokinase will form glucose 6-phosphate. So the phosphate is hanging off of the number 6 glucose in the number 6 carbon in the glucose ring. After this, you have phosphohexose phosphohexose isomerase, and this rearranges the molecule, and you get the formation of fructose 6-phosphate, which is represented here. So what happens is the number 1 carbon becomes part of a branch moiety at this point, and you convert the 6 member ring into a 5-member ring. Following that, phosphofructokinase 1 will take the fructose 6-phosphate and form, under a heavily regulated enzyme, taking another ATP and forming the compound here, which is an intermediate. And then this intermediate breaks with 2 phosphate groups attached to it, breaks with the use of aldolase, and that creates a glyceraldehyde 3-phosphate, which is right here, and the other molecule is dihydroxyacetone phosphate here. Then triose phosphate isomerase is an enzyme, and that creates 2 of these molecules here. Glyceraldehyde, triose phosphate isomerase occurs at this point, and you get the formation of 2 glyceraldehyde 3-phosphate molecules. So the first phase is through here. Second phase begins at this point when you have 2 molecules, and it works off of 2 molecules from that point on. You have inclusion of 2 phosphate ions at this point, reduction of NAD to NADH with the help of the enzyme glyceraldehyde 3-phosphate dehydrogenase. That forms the molecule 1,3-bisphosphoglycerate, which is represented here. And then 2 ATPs result from this. This is 2 phosphates attached to it, and one of these is hydrolyzed off, and you get 2 ATPs. So there's some energy that is produced at this point. You get from this 3-phosphoglycerate, which is here, and then another enzyme, phosphoglycerate mutase, produces a 2-phosphoglycerate. From that, with loss of water, you get a phosphoenolpyruvate, which is this molecule here, and then pyruvate kinase uh, produces 2 ATPs, and you get at the end of the process 2 molecules of pyruvate. So there's 1 molecule of glucose resulting ultimately in 2 molecules of pyruvate plus 4 ATPs, 2 here and 2 here. We will be going into further detail in these forthwith. So in phase 1, there are 2 molecules of ATP used prior to cleavage into 2 3-carbon units. In phase 2, inorganic phosphate produces 1,3-bisphosphoglycerate. 4 molecules of ATP are produced in the second phase, with a net of 2 ATPs, as 2 molecules of ATP were used in the first phase. So you get 2 molecules produced and 2 molecules of pyruvate as a result of this. Pyruvate has 3 possible fates. One of these is anaerobic conversion to lactate. Additionally, you can get fermentation conversion to ethanol and carbon dioxide, and then lastly, oxidation to acetyl-CoA, and ultimately CO2 in the citric acid cycle, or as you call it, the Krebs cycle. We'll be reviewing these shortly. Glycolysis also produces 2 molecules of NADH, and the overall energy is minus 85 kilojoules per mole, and that's an irreversible process. So let us talk about the individual steps in this pathway. The first of these in phase 1, which is the cleavage of glucose into 2 3-carbon moieties. The first step is phosphorylation of glucose, and this occurs with the help of the enzyme hexokinase, and the driving force is a magnesium ATP complex. You have a conformational change of hexokinase with an induced fit of glucose, which binds tightly, and the ATP is moiety is bonded at the number 6 carbon. The second step is the isomerization of glucose 6-phosphate to the 5-carbon fructose 6-phosphate with the formation of a methylene and hydroxyl group at the number 1 carbon. So the attachment of the oxygen ester to number 1 is switched over to number 2, and you get 5-carbon intermediate. The intermediate is an amine diol, where you get a double bonded entity with alcohol attachment that's present. So it would be carbon double bonded, and then the hydroxyl group present. This is an intermediate that occurs with the isomerase. The third reaction in this series is phosphorylation, which employs the enzyme, heavily regulated enzyme phosphofructokinase 1, or PFK1. Again, this is a phosphorylation occurs. This is at the number 1 carbon, and this entity works analogous to that of hexokinase, where you get liberation of the hydroxyl group and the phosphate group that is present at the terminus of the ATP molecule. The next step is cleavage of fructose 6-biphosphate to glyceraldehyde. This is a rather complicated process involving the active site of a lysine that attaches to the substrate at the number 2 carbon, and the carbonyl group then is attached by lysine here, picks up a proton for this carbon right here, and following that you get rearrangement, so you get a double bond of the lysine of the lysine nitrogen to the carbon here, with loss of the hydroxyl group, and then the process cleaves off of the number 4 carbon here, which breaks the molecule between number 3 and the 4 carbon. So the driving process occurs here, and you get oxidation at this point with cleavage of the carbon between numbers 3 and 4. And that's driven off of a lysine moiety that connects directly to the number 2 carbon and liberates these in the form of glyceraldehyde 3-phosphate, which is here, and the dihydroxyacetone phosphate here. The next reaction is dihydroxyacetone phosphate isomerizing to glyceraldehyde 3-phosphate. This mechanism is analogous to that of the second enzyme reaction in glycolysis, which is the phosphohexose isomerase reaction, where you get simple transposition of the oxidative potential of where you have oxidation at this carbon and reduction of that carbon. And there's no net change in the structure, but the redox potential is different between the two. And this is how you end up with 2 glyceraldehyde 3-phosphates. So when you look at the formation of these 2 3-carbon units, glyceraldehyde 3-phosphate, and you did backtracking of markings, this carbon, the number 1 carbon as we describe it here, is derived from either the number 3 or the number 4 carbon of glucose. This can either be number 2 or number 5 carbon from the original glucose, and this will represent either the number 1 carbon or the number 6 carbon of the original glucose. Analogous to the PFK reaction, you get then additional phosphorylation of the number 1 carbon utilizing oxidation of this molecule with NAD going to NADH in here. So you end up getting phosphorylation at both of these locations, number 1 and the number 3 formation. This is an energy conserving step, and the formation of this requires a cysteine molecule, and the cysteine molecule will react at this point and cause the negative charge to be accrued to the oxygen. That'll be the first point. So you have cysteine with a sulfur occurring, a sulfur carbon addict right here with the formation of hydroxyl group at this point. After that occurs, you can get this negative charge to form a carbonyl group attached to the sulfur, and this proton is released to NAD and reduces it to, so the proton here is what's occurring at this point here. So you end up getting the adduct of sulfur to a carbonyl group. Everything else remains the same. The next step is the phosphate, and the phosphate will attach at this carbon right here with release of cysteine. So cysteine is the adduct that helps in the part of the enzyme which helps us to proceed forward in the pathway. The next step is a phosphoryl transfer from 1,3-bisphosphoglycerate to ATP, and in this formulation you have this phosphate right here, ATP there, and the phosphoglycerate kinase, with the help of a magnesium cofactor, creates the reaction of transfer of the phosphate group to the terminus of ATP, and this will form the 3-phosphoglycerate as a consequence for this. The enzyme is also the same one that's used in gluconeogenesis. So this process can go in either direction. The enzyme works in this direction because it's coupled with the previous reaction. The next step is a conversion of 3-phosphoglycerate to 2-phosphoglycerate, and this is the enzyme phosphoglycerate mutase, which creates this transformation here. It's an energy-consuming step using magnesium as a cofactor. And in this particular operation, you have a histidine, which is used. The histidine has a nitrogen on it, and the histidine will cause will pick off this proton here and form a situation where phosphorylated histidine from another moiety will transfer its phosphate to the oxygen. So one histidine removes the proton. Another histidine will attach a phosphate group to this point here, and then at the same moment you would get the phosphate group here, which will reattach to that histidine that donated its phosphate to this point. So there are two histidines which are acting in concert, one to remove the proton, the other one to supply a phosphate and then remove this phosphate here. So you end up getting phosphate on the number 2 carbon as 2-phosphoglycerate, and that is the mechanism of this reaction. And the next step is 2-phosphoglycerate dehydration, the phosphoenolpyruvate. And this is a reaction that occurs with the help of the enzyme enolase. This is a situation where you get an alkene linkage here with elimination of water, and there's an intermediate that is stabilized by magnesium, and there is a energy-consuming step to this particular reaction. In this last step, the phosphoenolpyruvate has a very spontaneous, as judged by the free energy of negative 31.4 kilojoules, where phosphate is transferred to ADP to create ATP. There's magnesium with less possibility of manganese used as a cofactor, and this linkage here is reduced to get pyruvate here and remove the phosphate group. So we have at the end of the process glucose plus 2 NADs plus 2 ADPs and 2 phosphates, giving 2 pyruvates, 2 NADHs, 2 protons, 2 ATPs, and 2 water molecules. This is the end result of the process, and there is a net gain of 2 ATPs in this process, and the NADH can be employed for other entities as necessary in the Krebs cycle or for any oxidative phosphorylation method. And here's the net: 2 pyruvate, 2 ATPs, 2 NADHs. Glycolysis is totally regulated. This is aerobic metabolism, so it yields more ATP. Neoplasms consume more glucose than normal tissue. This is the basis of positron emission tomography, also called a PET scan. This is how you find tumor hotspots when you're looking for metastases in bone, for example, or other organs. There are glycolysis feeder pathways. Starch is a glucose polymer with alpha-1,4 linkages, which is hydrolyzed by amylase and secreted by the pancreas. Cellulose has beta 1,4 linkages, which are catabolized by microorganisms and ruminant animals. Endogenous glycogen can be catabolized by glycogen phosphorylase. You can generate glucose 1-phosphate from the terminus and converted by phosphoglucomutase to glucose 6-phosphate. A mutase transfers a functional group, and isomerase transfers bonds within molecules. As point of identification, maltose is comprised of two glucose molecules. Lactose is galactose and glucose. Sucrose is fructose and glucose, and all these are D-isomers. And fructose is converted to fructose 6-phosphate in the small intestine or fructose 1-phosphate in the liver. Ultimately, you can convert that to glyceraldehyde 3-phosphate, and under anaerobic conditions, pyruvate is converted to L-lactate. The net of these is 2 ATPs, 2 NADHs, and those are utilized by L-lactate formation. Lactate is stored in muscles and gives muscle pain after intense exercise. Over time, it is converted to in the liver to glucose. There are forms of industrial formation such as yogurt, Swiss cheese, pickles, and the like. Gluconeogenesis. Glucose is essential as a sole source of energy for the brain, nervous system, and embryonic tissues. This occurs in all living organisms. In humans, occurring mainly in the liver. The precursors are 3 and 4 carbon units mainly, and this is a separate pathway from glycolysis. When we look at glycolysis on this side and gluconeogenesis on the other side, we do have differences. So we're going with glucose. We covered this pathway that culminated in pyruvate down here. To go into the opposite direction, and we'll go through this process, pyruvate employs 2 ATP molecules with pyruvate carboxylase to get oxaloacetate. Phosphoenol pyruvate carboxykinase then takes 2 GTPs to GDPs, and from this you form phosphoenolpyruvate, and these are reversible reactions in common with the glucose pathway up until you get glyceraldehyde 3-phosphate. So the phase 2 is equivalent up to the point where you have phosphoenolpyruvate. There's one type of enzyme that goes to pyruvate and 2 enzymes that go in the opposite direction to get to phosphoenolpyruvate from pyruvate as a precursor. So once you get to the commonality of glyceraldehyde 3-phosphate, you produce dihydroxy acetone phosphate from that, fructose 1,6-biphosphate. There's a phosphatase, which removes the phosphate. You're back to fructose 6-phosphate. Fructose 6-phosphate with the same enzyme as in glycolysis to glucose 6-phosphate, and then the formation of glucose using glucose 6-phosphatase, which free phosphate, and then you get glucose coming back to that point. There are 3 bypass reactions of gluconeogenesis that avoid irreversible steps in glycolysis. The first of these, as stated previously, was pyruvate to phosphoenolpyruvate. These are 2 exergonic or heat-generating reactions that require 2 enzymes in 2 locations on the molecule. Pyruvate goes to oxaloacetate in mitochondria, and oxaloacetate to phosphoenolpyruvate in the cytosol. So these are 2 cellular locations involving the molecule. This is the first reaction which occurs where you have pyruvate. There is the addition of carbon via a bicarbonate donor molecule. This is pyruvate carboxylase, which is mediated through an adduct, which we will talk about in the next slide, utilizing ATP going to ADP and the formation of oxaloacetate. This opportunity exists given this structure to form an adduct with GTP, which forms a phosphorylated moiety at this carbon here with liberation. So the carbon dioxide is produced in this reaction, and you get a GTP, which is cleaved off. GDP comes off of that. Carbon dioxide comes from that, and you get phosphoenolpyruvate present with this bond going here and picking up this phosphate here. This part is very interesting, and we will see this right here. So we have the bicarbonate in solution. There is a biotin is the intermediary molecule, and there is a long biotinyl lysine tether from this point. So this is the enzyme which has site 1. There's lysine here at site 2, and the carbon dioxide is attached at this point. It has a tether which moves along from one site to another site, and then is transferred over to pyruvate, picking it up from this to oxaloacetate over here. So there is a movement of the carbon dioxide from one nitrogen and then moved over in tether form to the other one, and you get the pickup of pyruvate using this moiety right here. In this situation, you have ATP and GTP used in consecutive reactions. Lactate to pyruvate uses lactate dehydrogenase. The bypass reaction, fructose 1,6-biphosphate. The fructose 6-phosphate has hydrolysis of phosphate. Glucose 6-phosphate is then hydrolyzed to glucose, and you have one molecule of glucose to form it. You need 4 ATPs, 2 GTPs, 2 NADHs, and the overall free energy is negative 63 kilojoules per. So citric acid metabolites that we have convert to oxaloacetate. Also, gluconeogenic amino acids can be converted to glucose. For example, alanine and glutamic acid can be converted to glucose if necessary. Mammals cannot convert fatty acids to glucose. They cannot convert acetyl-CoA to glucose, but glycerol can be converted to glucose. And we will be discussing the pentose phosphate pathway in due time. That'll do it for this lecture. Thank you very much.